Clinical research. Before going to discuss about the clinical research, let's see a brief outline about the drug discovery and development process. Drug discovery and development process is the whole steps taken by the sponsor or the pharmaceutical companies in bringing a new medication to the public to cure a health alleviation or a disease. This process starts with an intention to develop a new drug or a new treatment procedure or a medical device to treat a disease in humans. So, in case of a drug, the drugs are available from different kind of sources like plants, animals, minerals, and some may be from the chemical synthesis process. Then the candidate drug isolated and purified from the sources. Then they are studied for their physicochemical parameters. Then after the candidate drugs are developed so as to produce particular therapeutic effect to treat a disease. This phase is known as the optimization process. It involves in the development of the candidate drug about its biological aspects. This is mainly done by the chemical, biochemical, and biotechnological advancements. Thereby, they improves the biochemical parameters and biochemical aspects of the candidate drug in such a way that it produces a desired therapeutic effect. After that, the whole information about the candidate drug, about its physicochemical parameters, that gives the preliminary information about the drug candidate. After gaining the preliminary information about the drug candidate, the sponsors steps further to evaluate the drug candidate in biological models. This is done to ensure that the development previously as made is practically producing the desired effects or not. Further, they proceed to test in the animal models and human models. At first, they conduct the biological evaluation in laboratory animals. This is known as the non-clinical study or the preclinical study. In this step, the drug candidates are evaluated for pharmacological and toxicological information in the animal models. After the successful evaluation of the pharmacological and toxicological information from the animal models, it proceeds to test in human models. It is known as the clinical study or the clinical trials. It involves through 4 phases by evaluating the pharmacology, the pharmacokinetic and pharmacodynamic information, and the efficacy of the drug and safetiness of the drug and dosage parameters, and any adverse reactions are evaluated through this phases. Then, after gaining the information from the biological evaluation, they seek approval for marketing the drug. So, throughout this process, the whole information and the data collecting every time is continuously updated into the databases for the statistical analysis to submit to the regulatory bodies. Thus, the drug discovery and development process involves an identification, developing, and evaluating the biological aspects, and then getting approved by the regulatory bodies to bring a new treatment or a new drug to the market. This is the whole discovery and development process. In this process, after gaining the preliminary information about the drug candidate to getting approvals by the regulatory bodies, the whole steps involved are known as the clinical research. This is about the clinical research as a part of drug development process. Clinical research is a part of drug development process. As we see in the previous slide, it involves in testing and assuring a compound for its diagnostic or therapeutic or prophylactic use in humans, and evaluates its rational, quality, efficacy and safety of the intervention. In the system of evidence-based medicine, a drug candidate should be tested and get approved, not only for a drug, for any treatment procedure or a medical device to use in humans as indication to treat a disease. These articles must be previously tested and get approved. The testing is done for making sure that the intervention is therapeutically safe and effective than already existing ones. So testing is mainly done for evaluating the drug or the treatment procedure or the device is safe to use and having effectiveness that is better than the already existing ones. The testing is sometimes reconsidered and may be terminated for its potential harms in subjects. The testing sometimes during testing the drug candidate in human models, they may produce suffocation and some unwanted reactions in the subjects. At the time, the testing procedures may be amended or terminated in case of lethal effects. So, the testing evaluates the farness and nearness of the results to intended purpose or outcomes. Thus, every testing evaluates useful, non-useful, positive, negative, relevant and irrelevant information about the intervention. The useful information is that the findings that supports the procedures or the methods of treatment. Some may be not useful in supporting the procedures. Some findings are positive about the procedures. Some may be negative. Some may be relevant and some may be irrelevant. But so this whole information and the findings are known as the data or the clinical trial data involved in clinical research. Clinical research involves through two steps dynamically. The first step is development of the scientific rationale and procedures for the intervention. The second step is to evaluate the scientific rationale is practically credible or not. Let's see about the first step. Drug development. Let's see about the first step. Development of scientific rationale and procedures for the intervention. After gaining the information about the drug candidate, the preliminary information is produced into the monograph. After that, from the preliminary information and by reviewing the previous scientific evaluations, the preliminary information is developed into a scientific proof document, which is known as the investigator brochure. It contains all the information about the drug candidate, the physico-chemical parameters, and the scope for biological activities or therapeutic indications. Procedures for the preclinical study are developed by reviewing and collecting the information about the treatment procedures in animal models, develops a preclinical study protocol. After that, the procedures for the clinical study are also developed from the investigator brochure, the clinical study protocol. So now see the second step involved in the clinical research, the site activities. After having this procedures for the intervention, then it has to be evaluated in biological models. Then the biological testing commences. The second step involves through two ways. One is testing activity, another one is regulatory activity. Coming to the testing activity, testing in animals, it is also known as preclinical studies or non-clinical studies or toxicological studies. This mainly involves in evaluating the pharmacology. It is about study of the pharmacokinetics and pharmacodynamics. And toxicological information. It is about the organ toxicity, the genetic and reproductive toxicity in the animal models. After successful evaluation of the pharmacological and toxicological information from the animal models, the whole information from the development process to the preclinical evaluation is submitted to the FDA through the investigational new drug application for seeking approval. After reviewing the investigational new drug application from the sponsor by the FDA may approve if it is satisfactory. Then the sponsor starts the clinical trials in human models. The clinical trials are conducted through 4 phases: phase 1, phase 2, phase 3, and phase 4. From the phase 1 to phase 3, the evaluation involves in the pharmacological aspects about the pharmacokinetics and pharmacodynamics and safety of the intervention, as well as the efficacy and also the dosing parameters and any other indications about the drug candidate are evaluated. After evaluation of these parameters, the whole information is submitted to the FDA through the new drug application seeking marketing approval. Then the FDA reviews the application, and if the FDA finds the evaluation has the benefits to treat a particular disease, then it approves. Then onwards, the post-marketing surveillance commences. It is also known as the phase 4 studies. Involves in the periodically reporting of the safety information to the regulatory bodies. These periodic safety reports are submitted to the Uppsala Monitoring Center. This is the safety regulating body in the world. It uses this information to minimize the medication errors. This is the whole activities involved in the clinical research. The biological evaluation starts with the animal studies, which are known as the non-clinical studies. It involves in the evaluation of the pharmacological and toxicological information in the animal models like rats, mice, rabbit, pigeons, dogs, monkeys, and etc. After the successful evaluation of the non-clinical data, then it proceeds to clinical evaluation in the human models. This involves the testing of drug candidate about its safety, efficacy, and pharmacology, and also the dosing parameters and any adverse reactions through 4 phases. After the successful evaluation of that, the seeks the marketing approvals. Throughout these phases, the whole information and findings are updated into a databases for the statistical development and representation. This information is known as the trial data or big data. This is the basic activity generally done at the site. There are also some regulated activity during these phases. Let's see at them too. The first regulated activity begins after the successful preclinical evaluation. The whole information from the development process to the preclinical evaluation is submitted through a new drug application to the FDA. Then after the clinical study starts, before setting the clinical study at the site level, an independent review board should be established by the physician to ensure the conduct of the trial is safe to the human subjects and to protect the rights of the trial participants. After successful evaluation of the information up to the phase 3, the whole information from the development process to the phase 3 evaluation about a drug candidate submitted to the FDA through new drug application seeking the marketing approval. After that, the phase 4 begins about the safety reporting. The periodically safety reports are submitted to the safety regulating body, the Uppsala Monitoring Center, for controlling the usage of medicines in safe manner. This is the representation about the clinical research and its activities.